I became fascinated with developmental biology as an undergraduate. I liked lots of aspects of biology, but then I was very fortunate. I had two professors who really got me excited about this particular research field. One of them was a lecturer who taught us about how flies and frogs and plants and humans develop, and so that really triggered a good response in me. And at the same time, I also had a supervisor who used to study human embryos, and he used to carry out these really careful measurements in the laboratory, looking at activity of human embryos at the very early stage in development. And at the time, the current thinking in science was that it was the most active cells that led to the most sort of successful embryos in establishing a pregnancy. But actually his work showed the opposite was actually true. It was what he called the quiet embryos that did really well at establishing a successful pregnancy after IVF. And so this really sort of changed the way people thought about IVF treatments, and also how we can sort of look at the quality of embryos that are most likely to lead to a successful pregnancy outcome. And so these two professors were very influential for me in making me excited about this area of work.
Well, I think, it's this a fantastic ability to actually discover new things. When we were doing a screen, actually using mouse embryonic stem cells to regenerate mice and to look at the consequences, and looking down the microscope one day, seeing what should be a normal, morphologically beautifully shaped embryo that just had gone completely haywire, and all the cells were folded on each other, and they'd lost their sense of direction and shape. And I realized that there's a single gene, a single protein that this embryo needs in order to actually set in place very precisely and reproducibly the early body plan of the embryo. So it was like, wow, there is one gene in our genomes that actually is responsible for orchestrating the very earliest stages of embryonic development. So it was really that's what kind of motivated me to say, well, how can one gene do this? Where is it normally made? Where is it expressed? How does it actually control cell-cell interactions and set in place, this early body plan of the embryo? It was a really kind of, also really piqued my interest.
I think it started back during my undergraduate studies. So embryology is not very much like a core subject. There were just few lectures about that, and I probably didn't understand much of it because it was so complicated and all these shape changes, and I found it extremely difficult to understand. And this is probably as well very fascinating, how different organisms from a single cell develop into such a complexity. So when I embarked into my master and PhD, I was interested as well in neuroscience, so I combined embryology and neuroscience. I studied the neural tube development. I work in the Great Ormond Street Hospital in London, where I did my PhD. And so I decided to go back in time in the embryology clock and study even earlier stages, so the stage of implantation, and specifically how the embryo transit from this blastocyst-looking structure into a successful implantation. So some type of research is more fundamental, and it's about general understanding and broadening knowledge. And of course, it's very important because even it shouldn't be all clinical focus. It should be discovery kind of driven more for the knowledge advancement that we can have in the field.